Thank you very much, Marie, for the very warm introduction. I sound very important with all that description, don't I? So it's my pleasure to be here today and to talk to you all about pancreatic cancer. And I'll get straight into it. Your tummies are full and you're well caffeinated, so you're in for the ride. So pancreatic cancer is a very tough disease, and I know that there are probably people in this room today who are all too familiar with these statistics, and the fact of the matter is that the statistics are still bad and they haven't changed much, only we've seen modest improvements over the past several decades. It's the 5th cause of cancer death, and only unfortunately 5% of people are still alive 5 years after they are diagnosed with this cancer. We don't see many early symptoms of the disease, and unfortunately most people are diagnosed with advanced disease where the disease has already spread out of the pancreas and into other organs. This is also really important as well because pancreatic cancer is on the rise, and there's some data to show us that between 1987 and 2007, that time period, there was only a 6% drop in mortality, so in the rates of death of this cancer. And this is way below what we saw death rates drop from other cancers, as you can see with lung cancer, 34% drop, bowel cancer, 47% drop. And even the rates of dropping of mortality with cancers overall was 28%. So we're still really lagging well below those rates of drops in death rates. And there actually are projections to suggest this is actually information we got from the US, but there is good evidence to say that the Australian population will follow. In 10 years, pancreatic cancer will be the 2nd highest cause of cancer death because we are seeing those increases in survival from the other common cancers, such as lung, bowel, prostate, and breast. So those cancers will continue to improve, and pancreatic cancer is projected to not improve at such high rates. So if you were diagnosed with pancreatic cancer today, and I will speak about this in very factual terms, you would have a 20% chance of being eligible for surgery, where they would remove your pancreas from your body with the tumour inside the pancreas. That is your greatest chance of beating the disease. If you then required chemotherapy after your surgery, which is what we call adjuvant chemotherapy, and don't be fussed by any of these fancy words, I'll explain what they all mean as we go along. There are a couple of different options here of what treatments you might be eligible for. Likewise, if your cancer was to return and you needed chemotherapy for where your cancer has spread to other parts of your body, there are then again a little collection of drugs that some of these are very toxic and be quite harmful. You may be eligible for some of these treatments. Now, why I wanted to put this up is just to really highlight the fact that if I was a cancer doctor and sitting in front of a patient for another more common type of cancer, I would have a list that would go over 5 or 6 slides of different treatment options that would be available to you as a patient. So this really brings us to, is this the best we can do in pancreas cancer? So what we do know is that patients need individualised treatment information. Every patient wants to know, will their cancer spread? Will they need to have chemotherapy? Are there any new drugs that might be available to them? And of course, will I survive? So we know these rates of survival and the incidence of pancreatic cancer is rising. What are we going to do about it? Really, we sought our information about we need to understand this cancer to really control, and ultimately the goal is to conquer it. And our research is working hard to attack where cancer starts in our DNA. So what is the connection between cancer and DNA? Well, DNA is the chemical that carries all the instructions that tell all the cells and ultimately then our organs and our body parts what to do. And when those instructions have mistakes in them, that's when cells may not function normally, and this can cause cancer. These cancer-causing changes, we call them mutations, and I'm going to use that word throughout the talk, can be inherited, so we can then get them inherited through our parents and our grandparents, but most of them are acquired throughout life, and some can be caused through environmental factors, such as chemicals, in the sun or through lifestyle choices such as smoking. But it does generally take the build-up of several different mutations to result in cancer. And that's a really important thing to remember for my next few slides. So the whole collection of our DNA in our body, we call that a genome. So what is this genome and why does it matter? So just like in the English alphabet, we have 26 letters, A through to Z. In the alphabet of our genome, there are 4 letters, A, C, G, and T. And these, just like a book, they make up the words to tell the story of our bodily function. So when we say things can go wrong in these stories, which can ultimately result in causing cancer, some even small changes in these letters of this alphabet can change what a word or sentence means in a story, the same as what happens in our genome. And you can see here, the first sentence, I've highlighted the O in boy. If we just switch out that letter in the sentence below that, it changes the meaning of the sentence completely. And this may only look like a really subtle change, but in the story of our genome, this is really important. We can also see things be added or deleted, and we can just even delete one letter in that same sentence. The letter O has been deleted in that same word, and you can see now that totally disrupts the entire meaning of that sentence. So we wanted to use this information in our genome to, as I said, attack where cancer starts in our DNA. And in particular, this was important in pancreas cancer if we were really going to make any headway in this disease. So here at the Garvan Institute, as Marie alluded to earlier, we started something called the Australian Pancreatic Cancer Genome Initiative. That's a really long word, and we just call it APGI because it's much easier to say, and it rolls off the tongue a lot easier than having to repeat the whole line. What we wanted to achieve with this initiative, it was a big consortium of different research groups around the country, we wanted to apply genome sequencing technology, so looking at new ways to interrogate these DNA inside our cells. We wanted to do this across a really large group of pancreatic cancer patients across the whole country. We recruited patients before they had their surgery, so thinking back to my slide where that was 20% of people that were eligible for surgery. We also wanted to collect information about previous medical history, risk factors such as if they smoked or not throughout their life, tumour features, and importantly, their family history. We were going to use this information to identify all the changes in pancreatic cancer cells, to ultimately understand them and to understand the different forms of pancreatic cancer and to find new ways to control them. This was an enormous project, and the data that we found during this study was made freely available to the research community. We didn't see the point of putting in so much effort, getting the patients involved, doing all this analysis to just keep it to ourselves. So this information that we have, that I will present to you in a few slides' time, is freely available for other researchers to access through a data portal on the internet. And this has been a coordinated effort in collaboration with a lot of other countries. This is an Australian-wide project. We have lots of different hospitals, lots of different research institutions involved. And likewise, this is being done on a global scale. This is a very busy diagram, a map of the world, but it just shows you the scale of this type of project and how many countries and how many different cancers were being looked at in this new way. So what have we learned? We started doing this in 2009, and we looked at 150 patients first. So we had to start slow and recruit patients before their surgery, and we got the first 150 patients, and we looked at their data, and we found over 2,000 underlying genetic faults, so genetic mutations, in these pancreatic cancers. And this is a bit of a busy graph, but we found some, these are all the lines of all the different genetic faults we found, some really high ones that are going right to the top of the graph, and then this long, what we call the tail of the graph, where these all look very similar. Most of these that we found were unique to individual cancers, and there were so many of these mutations, it's really difficult to know which of them were actually important. So that led us to our next study, where we knew we had to look at this even deeper. We needed to use newer technology and really find ways to look in a more detailed way at these genomes of these tumours. So the second time around, what we did, we had a detailed map of the entire genomic structure. And we found that tumours have these unstable regions hidden inside their genomes, and that there were actually existing classes of chemotherapy drugs used to treat other cancers in clinics available at the time that actually may be used to target patients that had these specific unstable areas. And this is what they look like. This is what we look at when we're in the laboratory and we have all of the data on an individual tumour. We can't look at these 2,000 different mutations on every patient because that would take us 100 years. So we draw diagrams of them that in a summary way show us what a patient's tumour looks like. So this is a real patient here, a real patient's tumour. And the letters of our alphabet are around the outside of the circle here. And if we look at this up close, you can see that there's looks like a bit of an earthquake. There's little blips on a radar that sit along this line here. And each one of those little blips represents a specific, unique individual fault inside that tumour. So you can see how many of those there are, and you can see every line, every blip on the radar represents something that's going wrong in that tumour. So this led us to our next phase of research. So we know pancreatic cancers all seem to be quite different. We saw so many mutations, we didn't know which ones were important. We know that they have these unstable regions hidden inside their genomes. So that then led us to look at it in a bit of a different way. We wanted to take all the information we had and look at it by using different analysis tools and look at it lots of different ways. So we here examined the whole genome, so the complete genetic makeup, and this was really the most comprehensive view we had of pancreatic cancer so far. And what we actually saw very strikingly in this big group of patients, over 400 patients, that pancreatic tumours could be grouped into 4 different subtypes, and each of these had their own unique genetic makeup and distinct behaviour. So when I say distinct behaviour, they all responded to treatments differently, and they all grew at distinctly different times. So what this was all leading us to, which we always had the hunch initially that this was going to be the case, that pancreatic cancer is more than one disease. So if pancreatic cancer is not one disease but many, this diagram here represents the genetic diversity of pancreatic cancer. What we're really dealing with, and I call this a bit like a licorice all-sort lolly. You have this straight line but with lots of different colours made up inside, and each of these individual colours probably represents an individual subtype of pancreatic cancer. And this is a really complex problem, because what this probably means that each of those individual colours probably needs its own targeted chemotherapy treatment that's exact to that individual genetic fault. So all of this research is breakthrough, and there's so much data, and it sounds so interesting, but what does this really mean for patients? This is probably the #1 question I get with my interaction with the patient community is, you know, all this fancy DNA stuff, it's wonderful, it's exciting, I don't really know what it means, but what does this really mean for me? And we've spent a lot of time soul searching about what we're going to do with this information next. So we know more than ever. We know more about pancreatic cancer than we've ever known before. That's a really positive and exciting thing. What is also striking is that nearly all patients receive similar treatments, despite us knowing now scientific evidence to show the fact that pancreatic cancer, they have very genetically different types. So we're putting all patients into the one group, giving them all the same chemotherapy drugs, when we now know that these are very individual tumours. So what we want to do is use this information and try and have more accurate, personalised treatments what we really want to do is be on the front foot so that we know what the underlying biology of these tumours are before we start treatment, rather than knowing this after the cancer has already spread, which is unfortunately what happens all too often in pancreas cancer. So you've probably heard a lot about personalised medicine, and I do get, this is probably the second most common question I ask. I mean, one would like to think that seeing their doctor and having prescriptions prescribed to them and treatments prescribed to them, that is personalised medicine. I'm getting a personalised treatment for me, for my cancer. But this is really taking that one step further. And it's really about the ability to not just diagnose disease, but to provide treatments that are particularly suited to an individual patient on a genetic basis. So what we want to do with the information learned in this study is really lay that foundation for personalised cancer care. So as an example, what we would do, we take a cohort of people diagnosed with pancreatic cancer. We apply these genomic sequencing technologies to them at the time that they're diagnosed with their cancer. We analyse this data. We find that there are these unique genetic faults across different patients. We then take those patients that have the similar unique genetic faults and group them into their individual groups on the basis of the genetics of their tumour. We would then give them drugs or test them in clinical trials that are specific for those tumours. And we would then see better success with these types of clinical trials because we are matching the right treatments to the right patients at the right time. So I'm going to switch pace a little bit now and talk a little bit more about some family history, which I talked about in the first few initial slides, because we do know that prevention has been and probably will continue to be the best way to improve health. And we know that through reducing cigarette smoking decreases cancer, and there are lots of examples of this. So if we can prevent pancreatic cancer, is this an area that we should be focusing on as well? So we do know that 5 to 10% of pancreatic cancers arise from genetic predisposition. It means we are born with a genetic fault or something else in our family that predisposes us to getting pancreatic cancer. So this can be done a few ways. It can be from family history, so having at least one pair of first-degree relatives, such as siblings or a parent-child, who are affected by pancreatic cancer. Or there are what we call cancer syndromes. And there are some, and don't be fooled by any of these fancy terms, but some of these may already be familiar to people in this audience. There is a hereditary breast and ovarian cancer syndrome, and a couple of others that are more rare. The breast cancer one's probably the most common. So if we know 5 to 10% of pancreatic cancers arise from this genetic predisposition, what causes that? What is this genetic predisposition? Do we know that? So we know a little bit. We know that this chunk, there are some known genes that are known to cause this genetic predisposition. Only 3 at the moment. There are a couple of newer genes that are coming out that are really under a lot of investigation in different laboratories around the world. But unfortunately, most of them we don't know. We can't explain a particular gene for some genetic predisposition in families. And this is another area we're really focusing on here in our pancreatic cancer research. We've set up a project called the Australian Familial Pancreatic Cancer Cohort, another big long name, established in 2011. And the aims of this were really to further understand the risks of what we call familial pancreatic cancer. So families where there are at least 2 relatives, first-degree relatives, affected by pancreatic cancer. We wanted to also try and discover genetic and non-genetic causes of this family history clustering in families. And we also wanted to be able to offer something to people who are deemed at high risk. And there is a screening trial that is underway just next door here at St Vincent's Hospital. So we have made great headway with this project as well. And there's over 300 patients that have signed up to this study. And what we found in our cohorts of pancreatic cancer patients in general, the incidence of familial pancreatic cancer is 1 in 10, 9% to be exact, but 1 in 10, which is what all the international literature says as well. So Australian data is showing the same thing. There are a couple of key differences I wanted to point out. When we looked at the familial patients, so cancers that are explained by family history, and those that what we call sporadic. So there isn't a hereditary component to them that we know of. They just have arisen throughout the course of the person's life. So we know that these two groups, when we put them side by side, that people are diagnosed almost around the same age. 65.8 if you're in the family history group, 66 if you're in the non-family history group. What's really important is that the patients in the family history group were more likely to have another relative with another cancer, namely breast or endometrial cancer. This was one fact that we found that we were not expecting. The rates were much, much higher, almost double of the people who in their family, extended family members, had another cancer of this nature. Familial patients were less likely to be smokers, whereas patients that are deemed sporadic cancers were much more likely to be active smokers, 28%. They also, in the sporadic category, had higher cumulative tobacco exposure, so had been exposed to this as a cancer-causing agent throughout their life. So I'm going to wrap up that now, but in conclusion, I hope I've convinced you that what we call genomic information, so all of the data we've learnt by studying tumours of patients with pancreatic cancer, it's really providing new opportunities for the discovery and development of new targets for cancer treatment. There's a lot of work underway to understand what the full impact of these cancer-causing mutations is. 2,000 mutations per tumour. We've got to be able to prioritise those mutations in some way to know which ones are most important and which ones may not be as important. And I think what's really important about this study, and it's really our legacy to research when I'm long retired and people underneath me are now doing this research, continuing it on, we're really amplifying the impact of scientific progress because we're enabling the future to build upon what we've discovered by offering up this data openly to the research community. Of course, this work would not be possible without all of our contributors. So all of our colleagues on a scientific level that have contributed to this study, there are over 150 across Australia of them. All of our clinicians, all that they do for our patients and looking after our patients and their families in what is undoubtedly the hardest time in their life. Our funding bodies who provide the funds to enable this to happen. And of course, our participants and their families, and I'm sure there are some of you in this audience right now. You are the ones who teach, inspire, and humble us. If you're at all interested in pancreatic cancer specifically, we're having another meeting on the 18th of June, and there'll be more specific updates from the APGI, and everyone is welcome to check out the website and register if you're at all interested. Thank you.